Hello there everyone, and welcome to this Biology Mind A-level tutorial on transport across membranes. So just to recap, as usual, we learned about the simple and facilitated diffusion processes in our last session together. We looked at how simple diffusion is the net movement, remember that key word net, of particles down a concentration gradient, while facilitated diffusion involves carrier and channel proteins to transport polar molecules and ions in and out of the cell. You now understand that there are multiple factors which can influence the rate at which simple and facilitated diffusion can occur. So let's have a look at our learning objectives for today. So number 1, an overview of osmosis. Number 2, water potential. And number 3, isotonic, hypertonic and hypotonic solutions. So we're just focusing on osmosis today. Alright, so let's crack on with this then. So osmosis has a very, very similar definition to diffusion. So if you know diffusion, you know osmosis. The key things I want you to remember of osmosis is that it's regarding water. So it's the net movement, again, this word net, movement of water molecules through a semi-permeable membrane, another important word to remember, from a region of high water potential to a region of low water potential. Okay, so again, just like how we talked about in our last tutorial, the word net is really, really important because that describes the overall trend of movement of water molecules, which is down its potential gradient. Now, the reason why we say we rely on thinking about the trend of the general movement of the water molecules is because some water molecules might move the other way, but overall many water molecules are moving from high water potential to a low water potential. Only some will move the other way. So therefore the net movement of water molecules is in that direction from high to low, just like with diffusion. Now notice we use this word potential with osmosis rather than talking about water concentrations. We talk about water potentials. We'll go on to think about what that is later on. Okay, so aquaporins are special channel proteins here that facilitate the diffusion of water. So you can see that they fit into the phospholipid bilayer. They're this channel here. They are designed to have hydrophilic pores which allow water molecules to move through them. So remember, hydrophilic is water loving, from the Greek hydro water, philic love. So water loving pores, so that attracts the water molecules and allows the water molecules to move through these channels. So one thing to remember is that it's the concentration of water that equalizes as a result of osmosis, not the volume. So for example here, if we're talking about water molecules moving from here to here, you can see that between this and this, there's an equal concentration of water, but the volume is not equal, okay? So when we think about osmosis, we're not equalizing the volume of water, we're equalizing its concentration in a solution. All right, so for example, if we have some particles in this side of the beaker, no particles in this side of the beaker, if we want to make this side of the beaker have an equal concentration of water, not of particles, but of water compared to this side, then we're going to have to add more water to this side to make this a higher water concentration, but also a higher volume. And therefore it's going to be equal to this concentration of water on this side of the beaker, but not volume wise, okay? Just concentration wise, it's going to be equal. So water moves down an osmotic gradient. So that's from high potential to low potential. So that's down an osmotic gradient. So when we talk about what water potential means, water potential is the pressure exerted by water molecules on a membrane, and it's measured in kilopascals. So when we talk about water potential, we're talking about the pressure exerted by water molecules on a membrane. So the units of water potential is this. Now this is the Greek letter called psi. Okay, so the units of water potential is psi with a small w next to it. So that's how we describe water potential in units. So psi with a small s is the solute potential, and it's a measure of the extent to which solute concentrations lower the water potential. Whereas psi with a letter P is the pressure potential, and it's a measure of the pressure that develops inside of a cell as a consequence of the inflow of water. So therefore, we can define water potential as the pressure potential plus the solute potential. Now, the key things for you to remember from this slide is don't worry about too much about the solute and the pressure potential for now. Just learn these definitions and learn how to define water potential using these definitions. All right, so those are the key things to take away. If I want you to take away one thing from this slide, it's just going to be that water potential is pressure potential plus solute potential. Now, the water potential of pure water at atmospheric pressure is 0. Adding solutes to water reduces the water potential. So the psi W has a negative value because we're reducing the water potential when we're adding solutes to it. Now, the more solute added to water, the more negative the water potential, okay? So basically, the less pure the water is, if you're going to put solutes into it, the less pure water is, the further away it is from 0, the more negative it is. Does that make sense? So the more solute added to water, the more negative the water potential of pure water becomes. So in osmosis, water moves down a water potential gradient across semi-permeable membranes. We've already got that definition in our heads. So water's moving from a region of high water concentration, so i.e. less negative psi W, to regions of low water concentration, so more negative psi W. So remember, something that is more negative, so for example, let's take the number -50. That's more negative than -10, for example, but they're both negative values. Now, this is what I would say is a higher water concentration because it's higher than -50. This is a lower water concentration because it's lower than -10. So in this case, for example, if we had a membrane between these two solutions, which way would we get water moving? Well, we know that water moves from a region of higher water concentration, so from this less negative value, to the region of lower water concentration, to this more negative value. So this is our gradient, and so this is the way that our water molecules are going to be moving. I hope that makes sense. Okay, so let's think about now our isotonic solutions and how we can vary these. So something that is isotonic has an equal water potential. So this is an isotonic example because this bit is similar water potential to this bit inside the cell, okay? So we say this is isotonic. So this is the same water potential within the cell as it is outside of the cell. So we have this nice equilibrium achieved here. Now, in a hypertonic solution, all this means is the outside of the cell, so the solution around the cell is more hypertonic, i.e. it contains less water. So what's going to happen is there's a lower water potential. So out here, there's a lower water potential. In here, in the cell, there's a higher water potential. So what does that mean? Which way is water going to travel? It's going to travel out of the cell, down the concentration gradient. So what does this lead to? It leads to sort of shrinking of the cell because water is moving outside of the cell. Now, on the opposite side of things, a hypotonic solution is when there's a higher concentration, higher water potential outside of the cell compared to inside of the cell. And therefore, which way are we going to get water moving? We're going to have water moving into the cell. And this, if you can think about it, a cell is an enclosed space within a membrane, as we've learned in previous tutorials. So if we have water moving in, if too much water is moving in, our cell's going to start to get a little bit rigid here. Can you see it's filling up with all of this water that's moving in by osmosis? And ultimately it can burst. So this is the effect of putting a cell into a hypertonic versus a hypotonic solution. And my top tip here is to think about in each of those cases, where is the water potential highest and where is the water potential lowest? If there's a solution that is hypertonic, it's going to have a lower water potential. If there's a solution that is hypotonic, it's going to have a higher water potential. And therefore, this is all compared to the cell, okay? And therefore, water is always going to move down its potential gradient, as we found out in the definition, via the semi-permeable membrane. All right. So that completes our lesson for today. Well done for following through to the end, and I'll see you for our next session.